Welcome back to the playlist on nucleotide and nucleic acid catabolism. More specifically, this is the purine playlist where we talk about purine catabolism. In the last few videos, we've been talking a lot about urate oxidase and urate. More specifically, in the last video, we looked at urate properties and structure in organic and biochemistry and so forth. We're going to do a little bit of that today, but what we're actually going to do is focus more on non-enzymatic urate reactions. Okay. Now, in the last video, we talked about urate structural properties. We didn't talk about any reactions that it did other than autoionization with water. But what I mentioned in the last video at the very end of it was that urate actually has some protective properties against free radicals. So notice here, I've drawn one free radical. So this particular free radical that I've drawn, this is what we call a hydroxyl radical, okay? It's not a hydroxide because, as you notice here, it's one electron short of being a hydroxide anion. Okay, so it's a neutral molecule, but overall it's extremely reactive, and these free radicals like hydroxyl radicals and superoxides and hydrogen peroxide, these guys can be produced by things like respiratory burst if you're eating inflammatory foods. Okay, but here's the idea that I want to sort of drive home. Okay, and the idea is that these urate molecules, and specifically this is the deprotonated form. If you need help with urate oxidase and urate properties, I highly recommend you go watch the previous video. But this is the urate form. This is the deprotonated state. So here's the idea that I want to drive home. If you look at great apes, so up here these are in general what we call great apes. Some of them are what we call hominids and so forth, and humans are of course included. These are animals, okay, that lack, keyword is lack, urate oxidase. So what does that mean for these guys? Well, for these animals, it means that they don't have any way to catabolize urate. And so they're going to have slightly higher levels of urate in the blood than other animals. Okay, like these ones are shown down here. So what I have for you are basically the average lifespan for these animals. Okay, so I looked up one primate that is not classified as a great ape, and so it has a functional urate oxidase, a lemur. Its lifespan is about 20 years. Dogs have urate oxidase. Their lifespan is about 12 years. Cats have urate oxidase. Their lifespan is about 18 years. Okay, so all of these guys that I'm showing here are mammals. Okay, so they're sort of classified in the same group. But these up here, all these guys up here lack a functional urate oxidase. Notice one of them is 50 years, which is actually pretty decent. But notice once you start getting into the bonobos and the apes, and the chimpanzee and the gorilla, now you're starting to get up to 70 and 75 years. And obviously humans, some of them can live to be 120. So we're talking about average lifespans. So people can live a lot longer than these, and certainly chimpanzees and so forth can live a lot longer than 75 if they're kept in captivity. Okay, so why might it be that great apes, the higher level primates, why might it be that they could live longer? Could it be that because they lack a functional urate oxidase, could it be that the higher levels of urate that they produce and get into the blood have some protective properties? It's a well-established fact that radicals cause aging. If you eat lots of inflammatory foods like dairy is an inflammatory food. Gluten's an inflammatory food. So anything that promotes inflammation, okay? If you're eating foods that promote inflammation, then you're getting hydroxyl radicals. You're also getting superoxides. You're getting superoxides and hydrogen peroxide. It's a well-established fact that these things produce aging. Let me circle that in red because it's bad. These produce aging properties. Okay, so is it possible that because humans and higher level primates lack a urate oxidase, that the urate in some way provides a protective effect from radicals? And hopefully in this video you'll kind of see the proposed mechanisms of how they do that. So through things like respiratory burst and obviously other types of metabolism, you can boost hydroxyl radicals. Well, it turns out that because urate has some pi electron conjugation, you can actually tie up a radical in that electron conjugation, and it can render the electron significantly less reactive, and then the species that you get at the end can be excreted. So for example, I'm going to draw the mechanistic steps in green. The hydroxyl radical can essentially couple with the urate ring. So then one of these electrons can come out here and couple, and then this one can kick onto this carbon right there. Okay, and that obviously generates this molecule right here. So notice we've now tied up the hydroxyl radical onto the urate ring. And now we have, of course, we have the lone electron, the radical electron, in the urate type of ring. And then, of course, this radical electron can, be bounced around the ring and so forth. Okay, and you can draw a ton of radical resonance structures where you put the radical electron on different atoms. Okay, you can certainly do that. But the point I want you to see is that, number one, you take a very reactive and unstable hydroxyl radical, and then you tie it up to the urate nucleus, and then you actually render the radical electron astronomically less reactive. Okay, so now the radical is significantly less reactive, and it doesn't do much. And then this molecule right here, this proton should be gone. Let me erase that. So let me erase that. Okay, there we go. And so now the radical electron is significantly less reactive, and it can't cause really any damage. And in the process, you of course tie up the hydroxyl radical as now a hydroxyl group on this urate nucleus. So this would be sort of a mechanism as to how it would exert its anti-inflammatory properties. And specifically, normally what we're considering is this carbon right here as the electron donor, the radical electron donor. This is a highly activated carbon in the urate nucleus. And part of the reason for that is because you can actually tie up the radical electron, as you can see, in multiple atoms here in the urate nucleus. And of course, you can draw more radical resonance structures. Okay. Another example of a radical that you can have floating around is a superoxide. So this molecule right here that I've drawn is called a superoxide. We're actually going to see some interesting shift-base chemistry here. So once again, what can happen is the radical electron, which is shown right here, it could couple with the urate ring. So again, I'll draw the radical electron, and I show the radical electron right here on this carbon. So now what I have on the urate ring is a peroxy linkage. Okay, this group right here, when you have these two oxygens bound together and to something else, this is called a peroxy group. This is called a peroxy group. Okay, and of course, just like in the last case, I can sort of shuffle that electron around. So notice this electron could come over here, right, and you can force the radical electron onto this carbon. It doesn't really matter for these purposes, but the idea is that I can have shift-base formation now. So you're assuming, there's a water or some kind of activated water here in the solution around the urate. So what I can get is essentially a shift-base formation. Right? I can get a shift-base formation, and that's going to cause loss of a leaving group. Well, the leaving group would be the peroxy species, but it can actually come and abstract the proton from that nitrogen, right? And in the process, of course, I'll do this in another color, the peroxy species would pick up a proton from solution, okay, possibly from a hydronium, or it could be from a water, okay? And that generates, it can generate a variety of really strange urate type of species. But the idea from that particular reaction is we generate a bad molecule, which is called hydrogen peroxide. Okay? Well, it seems we're well looked after, because normally when we talk about catalase, we're talking about intracellular catalase. But it turns out that there's actually also an extracellular catalase to protect us from any hydrogen peroxide species that might be produced. So it basically requires 1.5 hydrogen peroxides, and it catalyzes the conversion and deactivation of them to these products as shown right here, which are molecular oxygen and water. Okay? So you can hopefully see at the end of this video, you can see the many protective effects that we get from, number one, the extracellular catalase, but of course the urate molecule itself. And maybe on some level, this, if I can find my mouse, this could explain perhaps one of the reasons why hominids and great apes like humans live longer than many other mammals. So hopefully this video gave you a little bit more intuition on urate. See you in the next video. In the next video, we're going to look at the catabolic pathway for GMP. See you in the next video.